Welcome to my channel XY Biology Academy. Imagine placing a bacterial smear on a glass slide and looking through a microscope. Within seconds, tiny cells that are completely invisible to the naked eye become visible. But have you ever wondered how a bright field microscope actually makes those cells visible? And why is it called a bright field microscope in the first place? The name itself gives us the answer. In this microscope, the background, or field of view, appears bright, while the specimen appears darker than the background. This difference in brightness creates contrast, allowing us to see the specimen. That's why it is known as a bright field microscope. Now let's understand the working of a bright field microscope step by step using its ray diagram. The process begins with the light source, usually an LED or a tungsten lamp, located at the base of the microscope. The light emitted from the source now travels upward, but before it reaches the specimen, it passes through the iris diaphragm. The iris diaphragm is a thin circular structure made up of overlapping metal blades that form an adjustable opening in the center. By increasing or decreasing the size of this opening, it controls how much light is allowed to pass through the microscope. In simple words, the iris diaphragm acts like a light controller. It doesn't focus light or magnify the specimen. Its only job is to regulate the amount of light entering the optical system. Now let me ask you a question. If the iris diaphragm can allow more light to pass through, why don't we simply keep it fully open all the time? Wouldn't more light always produce a better image? The answer is no. If too much light passes through the specimen, the entire field becomes excessively bright. As a result, the specimen blends with the background, reducing contrast and making fine details difficult to distinguish. On the other hand, if very little light enters the microscope, the image becomes too dark to observe clearly. So, the iris diaphragm is adjusted to achieve the right balance between brightness and contrast, allowing the specimen to be seen clearly. After passing through the iris diaphragm, the light reaches the condenser. The condenser is not a single lens. It is a system of two or more convex lenses placed beneath the stage. These lenses are specially designed to collect and direct light efficiently. Now think about a light bulb for a moment. The light coming from a bulb spreads out in every direction. If we allowed this scattered light to fall directly on the specimen, the illumination would be uneven, and the image would lose sharpness. This is exactly where the condenser becomes important. The condenser collects these scattered rays and refracts them so that they converge into a bright, focused cone of light. This concentrated beam is then directed precisely onto the specimen, ensuring that every part of it receives uniform illumination. You can compare it to using a magnifying glass under sunlight. Sunlight normally spreads over a large area, but the magnifying glass gathers those rays and concentrates them into one small, bright spot. The condenser works on the same optical principle. The focused light now passes through the specimen mounted on the glass slide. This is the most critical part of the entire mechanism. Now think about this. If every ray of light passed through the specimen without changing in any way, would you be able to see the specimen? The answer is no. If all the light passed through equally, the specimen and the background would appear identical. There would be no contrast, making the specimen practically invisible. Fortunately, biological specimens interact with light. Different parts of the specimen absorb, scatter, refract, and transmit light differently depending on their thickness, density, and chemical composition. For example, thicker regions absorb or scatter more light, so fewer rays reach the objective lens from those areas. These regions therefore appear darker. In contrast, thinner or less dense regions transmit more light, allowing more rays to reach the objective lens, so they appear brighter. This difference in light intensity between different parts of the specimen creates contrast, and this contrast is exactly what allows us to distinguish the specimen from the bright background. That is also the reason this microscope is called a bright field microscope. The background remains brightly illuminated, while the specimen appears darker because it blocks or scatters part of the transmitted light. Now let's continue following the light. After emerging from the specimen, the transmitted light enters the objective lens. If someone asks you which lens is the most important in a microscope, your answer should always be the objective lens. Why? Because the objective lens is responsible for producing the first magnified image of the specimen. It collects the transmitted light rays and bends, or refracts, them so that they converge to form a real, inverted, and enlarged image inside the microscope tube. Now here's another question. Why is this image called a real image? Because the light rays actually converge and meet at a specific location. In other words, the image is physically formed inside the microscope and could even be projected onto a screen if the optical system allowed it. However, this real image forms inside the microscope tube, so we cannot observe it directly with our eyes. Instead, this real image now becomes the object for the next lens, the ocular lens, also known as the eyepiece. The eyepiece acts like a simple magnifying glass. It takes the already enlarged real image produced by the objective lens and magnifies it even further. Unlike the objective, the eyepiece does not produce another real image. Instead, it produces a virtual image, which is the image our eyes finally perceive. This means that when you're looking through the microscope, you are not directly observing the specimen. You're actually observing a virtual image created from a real image, which itself was formed by light passing through the specimen. Now one final question. Which lens contributes more to image quality, the objective lens or the eyepiece? The answer is the objective lens. The objective determines most of the microscope's resolution and image quality, while the eyepiece mainly provides additional magnification. That's why high-quality objective lenses are much more important than high-power eyepieces. Finally, let's calculate the total magnification. The total magnification is simply the product of the magnification of the objective lens and the magnification of the eyepiece. For example, if you're using a 40 times objective lens together with a 10 times eyepiece, then the total magnification is 40 times 10 equals 400 times. This means the image appears approximately 400 times larger than the specimen's apparent size when viewed with the unaided eye, although magnification alone does not increase detail unless the microscope also has sufficient resolving power. Now let's look at the advantages of the Bright Field Microscope. It has a simple design, making it easy to use, especially for beginners. It is relatively inexpensive compared to advanced microscopes, making it the most common microscope found in schools, colleges, hospitals, and research laboratories. It provides natural color images when stained specimens are used and offers high-resolution images for observing stained bacteria, tissue sections, blood cells, and many other biological samples. However, it also has some limitations. The biggest limitation is that unstained living cells are almost transparent, so they produce very little contrast and are difficult to observe. In most cases, specimens need to be stained, but staining usually kills the cells, making it impossible to study living organisms. In addition, the microscope cannot reveal the fine internal details of transparent living cells as effectively as phase contrast or differential interference contrast microscopes. So, the Bright Field Microscope works on a simple principle. Light is focused onto the specimen, different parts of the specimen transmit different amounts of light, and the objective and eyepiece lenses magnify this transmitted light to produce a visible image. The background remains bright, while the specimen appears darker, giving this microscope its name, the Bright Field Microscope. Thanks for watching. Like this video and subscribe to my channel.